हेलो फ्रेंड्स। टुडे हम एनसीईआरटी बायो 11 का चैप्टर नंबर 8 सेल यूनिट ऑफ लाइफ रीड करेंगे एंड लर्न करेंगे। चैप्टर शुरू करने से पहले जान लेते हैं, याद रखना, जिसने अपने मन को वश में कर लिया है, जिसने अपने मन को वश में कर लिया है, उसकी जीत को देवता भी हार में नहीं बदल सकते। उसकी जीत को देवता भी हार में नहीं बदल सकते। याद रखना। इसलिए अपने गोल को पाते रहना है। नेवर गिव अप मेरे दोस्त, नेवर गिव अप। चलो अब स्टार्ट करते हैं। चैप्टर नंबर 8 सेल द यूनिट ऑफ लाइफ। व्हाट इज अ सेल? Unicellular organisms are capable of independent existence and performing the essential function of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope reveal all the structure details of the cell. Then next, cell theory. In 1838, Matthias Kelden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form a tissue of the plant at about the same time. Theodore Skevan in 1839, a British zoologist, studied different types of animal cells and reported that cell had a thin outer layer which is today known as the plasma membrane. He also concluded based on his studies on plant tissues that the presence of cell wall is a unique character of the plant cells. On the basis of this, Skevan proposed the hypothesis that the bodies of animals and plants are composed of cells and products of the cell. Skeldon and Skevan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cell divide and new cells are formed from pre-existing cells, that is, omni is cellula. He modified the hypothesis of Skeldon and Skevan to give the cell theory a final shape. Cell theory as understood today as: first, all living organisms are composed of cells and products of cell. Second, all cells arise from pre-existing cells. An overview of cell. You have earlier observed cell in an onion peel and or human cheek. Cells under the microscope. Let us recollect their structure. The onion cell, which is the typically plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cell of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nuclei are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cell. Various chemical reactions occurs in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, the Golgi complex, liposomes, mitochondria, microbodies, and vacuoles. The prokaryotic cell lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, that is, chloroplast in plants and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelles called centriole, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasma, the smallest cell, are only 0.3 micrometer in length. These diagrams represent these various cells in NCERT books. Please see. While bacteria could be 3 to 5 micrometer, the largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Then next point is prokaryotic cell. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organism. They are generally smaller and multiply more rapidly than the eukaryotic cell. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, that is rod-like, coccus, spherical-like, vibrio, comma shape, and spirillum, that is spiral shape. The organization of the prokaryotic cell is the fundamentally similar, even though prokaryotic exhibit a wide variety of shape and functions. All prokaryotes have a cell wall surrounded the cell membrane. The fluid matrix filling this cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome, that is circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to the such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with the foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cell, except for ribosomes. Prokaryotes have some things unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotic. They are essential in folding of cell membrane. Then next point of NCERT, that is cell envelope and its modification. Most of prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layer structure, that is the outermost glycocalyx followed by the cell wall and the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of difference in the cell envelope and the manner in which they respond to the staining procedure developed by Gram. That is, those that take up the Gram stain are Gram positive, and then others that do not are called Gram negative bacteria. Glycocalyx differs in the composition and thickness among different bacteria. It could be the loose sheath called slime layer in some, while in other it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membraneous structure is semi-permeable in nature and interact with the outside world. This membrane is similar structure to that of the eukaryote. A special membrane structure is the mesosome, which is formed by the extensions of plasma membranes into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in the cell wall formation, DNA replication, and distribution all to daughter cell. They also help in the respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotic like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contains pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of the three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made by a special protein. The fimbriae are small bristly-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in the stream and also to the host tissue. Then, next point is ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 50 nanometer by the 20 nanometer in size and are made of two subunits, that is 50s and 30s unit, which when present together form 70s prokaryotic ribosome. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of polysomes translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cell are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophycin granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotic includes all the protista, plants, animals, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cell possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and a large central vacuole which are absent in animal cell. On the other hand, animal cells have centrioles which are absent in almost all plant cells. The given diagram in NCERT shows the plant cells and animal cell. Please see carefully. Next point is cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950th century. Meanwhile, chemically studied on the cell membrane, especially in human red blood cells (RBCs), enable the scientists to deduce the possible structure of the plasma membrane. These studies shows that the cell membrane is composed of lipids that are arranged in bilayer. Also, the lipids are arranged within the membranes with the polar head toward the outer side and the hydrophobic tails toward the inner part. This ensures that the non-polar tails of the saturated hydrocarbons is protected from the aqueous environment. The lipid components of the membrane mainly consist of phosphoglycerides. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of the protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipid. Depending on the S of extraction, membrane proteins can be classified as integral or peripheral. Peripheral proteins lie on the surface of membrane while the integral proteins are partially or totally buried the membrane. The following diagram shows fluid mosaic model of plasma membrane. Next point, an improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intracellular junctions, secretion, endocytosis, cell division, etc. Next, one of the most important function of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solute may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to the lower. Water may also move across the membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is, from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized. ATP is utilized and cell wall is called active transport. Example, as you sodium potassium pump. A non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protect the cell from mechanical damage and infection, it also helps in the cell to cell interaction and provides barrier to undesirable macromolecule. Algae have a cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell mature, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by a plasmodesmata which connect the cytoplasm of neighboring cells. सो फ्रेंड्स, आज इतना ही. आप प्लीज़ एक बार फिर से रिवाइज कर लीजिए और एक बार फिर पढ़ लीजिए, क्योंकि नीट में और एम्स में हमें मालूम है कि 90% पेपर एनसीईआरटी बेस होता है, इसलिए हमें इसे पढ़ना ही है. आप बार बार सुनिए. सुनने से क्या होता है, हमें चीज़ें जल्दी याद हो जाती है. इसलिए हम नेक्स्ट एंडोमेम्ब्रेन सिस्टम पढ़ेंगे. तब तक के लिए प्लीज़ आप रिवाइज कर लीजिए. चलिए फ्रेंड्स, मेरी ये कोशिश आपको ज़रा से भी पसंद आई हो तो प्लीज़ सब्सक्राइब ज़रूर करना, इससे मुझे मोटिवेशन मिलेगा. और हाँ, लाइक्स और शेयर ज़रूर करना, इससे आपके फ्रेंड्स को मदद मिलेगी. और फ्रेंड्स, यह वीडियो आपके सारे फ्रेंड्स को शेयर करना, जिससे उनकी भी मदद होगी. सो फ्रेंड्स, जय हिंद, जय भारत. मिलेंगे अगले वीडियो में.